Two people can eat exactly the same amount of sugar, yet their bodies handle it differently. While one person stores it as fat, yet another stays lean, seemingly without effort. To find out why, researchers recently screened stool samples from 472 donors. They were looking for specific lactic acid bacteria capable of processing dietary sugar inside the gut. This chart plots the abundance of a specific microbe called Streptococcus salivarius across those donors. The data shows a clear divide. Lean subjects consistently harbor high levels of this bacterium, but in subjects with a body mass index over 30, those bacterial counts fall toward 0. There is a direct inverse correlation between the amount of S. salivarius in the gut and a person's body mass. This microbe is a picky eater. If you feed it glucose or fructose, it ignores them. It only produces these metabolic byproducts when it has access to pure sucrose, or table sugar. There is a biological irony here. This organism specifically evolved to process the exact type of refined sugar driving the modern Western health crisis. For those who stay lean on a high-sugar diet, the cause might not be willpower alone. It may depend on harboring this microscopic sugar interceptor. The effectiveness of S. salivarius comes down to its location. While most well-known gut microbes reside in the colon, this bacterium colonizes the small intestine. Normally, enzymes break sucrose into simple sugars that pass the intestinal wall, promoting fat storage. But living here, S. salivarius intercepts the sugar first. It creates a biological bypass, converting it into heavy, indigestible polymers called exopolysaccharides. Human digestive enzymes like amylase cannot break the bonds of these new polymers. This allows the sugar mass to pass through the small intestine intact. By occupying this anatomical choke point, the bacteria reroutes dietary sugars away from your bloodstream and fat cells. That intercepted sugar then drops into the colon. Here, it becomes a specialized fuel source for fiber-degrading bacteria like bacteroids. Through cross-feeding, colon bacteria ferment the EPS into metabolic signals called short-chain fatty acids. These messengers lock into host cell receptors, GPR41 and GPR43. Activated, they signal the body to increase energy expenditure, suppress fat, and release fullness hormones. In mouse studies, those fed specific EPS polymers gained less weight and improved their cholesterol levels. Their bodies even began browning their white fat, effectively turning storage units into energy-burning furnaces. We know this circuit is essential because when researchers removed the receptors or the bacteria, the anti-obesity effect disappeared entirely. This bypass mechanism transforms raw dietary sugar into a biological command to burn fat. The process is a complete metabolic loop. S. salivarius intercepts sugar in the small intestine, converts it to fiber, and sends it to the colon to signal fat burning. Lacking this bacterium is like having a stuck open valve. Without the microbial blockade, a torrent of unmitigated sucrose rushes into the system, making it difficult to resist the metabolic impact of a modern diet. In the near future, S. salivarius levels could serve as a biomarker to identify individuals who are most vulnerable to the effects of high-sugar diets. Researchers are now developing symbiotics that combine live S. salivarius with targeted prebiotics to help regulate how a patient's gut processes sugar. You can support this system today by eating a diverse range of fibers. This nurtures the bacteroides in your colon, keeping them ready to process those sugar polymers when they arrive. However, this biological bypass has limits. Even a healthy population of bacteria can be overwhelmed by a massive overload of sucrose, so mindful intake is still necessary. Weight management is a collaboration between our dietary choices